Darwin still reigns.
Darwin reigns. The theory of evolution is an absolute paradigm that holds all intelligence captive. There are many people who are unwittingly trapped by this deception, but these people will soon be free, because now Darwin's powerful illusion can be rigorously and scientifically falsified.
What we have just heard are not the words of a pseudoscientist, but the words of Professor John Sanford, one of the most important geneticists in the world. But how did John Sanford, a world-renowned scientist, come to formulate a theory that contradicts decades of study and experimentation by his colleagues? To fully understand the revolutionary scope of this theory, we need to take a step back and start from the beginning of this story. Today we will talk about the theory of genetic entropy, a complex theory. Complex, fascinating, but also extremely divisive. As you can easily imagine, Professor Sanford's theory has sparked very strong reactions in the scientific world. The denial of the validity of neo-Darwinian theories has brought him fierce criticism and led part of the mainstream scientific community to distance itself from him. But what did the professor discover that was so important that it led him to reject years of research and studies? John Sanford graduated in 1972 from the University of Minnesota, where he later specialized in plant genetics. From 1986 to 1998, he was an associate professor of horticultural science and was also an honorary associate professor of botany at Duke University. Over the years, the professor has published more than 70 scientific papers and has registered as many as 32 patents. At Cornell University, Sanford and his team developed the biolistic particle delivery system, the so-called gene gun. Gene gun is a method for introducing foreign DNA into a cell, with the goal of creating transgenic organisms. This is currently the most widely used direct transfer method for transforming plant cells. The professor has been awarded the prestigious Distinguished Inventor Award twice for the significance of his research. In addition to this, the professor has founded two biotechnology companies, Sanford Scientific and Biolistics. With this quick introduction certifying the professor's expertise, let's get to the heart of the discussion. And since Sanford is a geneticist, we can't help but start by talking about the genome. The genome is the complete set of an organism's genetic material, contained in the nucleus of its cells and organized into structures called chromosomes. In the case of humans, the genome is made up of about 3 billion pairs of DNA bases, which contain the information necessary for the construction and functioning of the body. This information is encoded in the form of genes, which are specific segments of DNA responsible for the production of proteins and other functional molecules.
The genome can be compared to a vast collection of encyclopedias that serves as a detailed manual for the construction of both a single human cell and the entire human body. Although it may seem similar to a computer program or an operating system, the genome is actually much more sophisticated and advanced than any technology developed by humans. Every year we discover new wonders about it, realizing that it is even more astonishing than we had previously imagined.
The Human Genome Project was one of the most ambitious scientific efforts in history. Officially launched in 1990 and completed in 2003, the Genome Project aimed to map all the genes of human beings and determine the sequence of the chemical bases that make up human DNA. This project involved scientists and laboratories from all over the world and represented a turning point in genetics and molecular biology. The genome is at the foundation of everything we know and call life. But according to Professor Sanford, the genome, however, follows a universal law, a law that all of us deal with every day: entropy.
Entropy is a concept that is interpreted and defined in different ways, depending on the discipline. While physicists may have a technical definition based on thermodynamics, engineers or information scientists might see it from a different perspective. But at its core, entropy represents a simple, universal truth: the intrinsic tendency of everything to disperse, deteriorate, decay, and move toward an end. It's not an abstract concept or something far from our daily experience. It's something we observe and experience every day. Many recognize it as Murphy's Law, that idea that if something can go wrong, it will. Whether it's a new car that starts to show signs of wear over time, or a house that needs constant maintenance, entropy is that constant and pervasive force we struggle against every day in our lives.
If everything tends toward chaos, from our home to the outside world to the entire universe, does this universal law of disorder also apply to living beings and specifically to our genome? Let's try to simplify a concept, that of aging. In the body of a human being, we have about 3 new mutations every time a cell divides. This, at the moment, according to scientists, is the reason why all of us age. All our systems start to collapse because of this mutational process, which accumulates degenerative mutations in our genome. This is basically the reason why there is an upper limit to lifespan. But according to Professor Sanford, the problem is not limited to the individual, but is even bigger.
Decades ago, a well-known geneticist predicted that with just 1 mutation per person per generation, the degeneration and extinction of the human species would be certain. Recent discoveries show that the mutation rate is much higher, with over 100 mutations per individual in each generation. This implies that each individual carries about 100 mutations not present in their parents, and in turn he passes them on to his own children. With each generation, about 100 new mutations are added to the human genome, and this is a conservative estimate. It is believed that the mutation rate could be even higher. In the current scenario, each of us carries tens of thousands of negative mutations. The reality is that we are all mutants, and significantly, statistics show that about 2% of newborns have obvious congenital defects. This percentage represents only the tip of the iceberg. Most mutations are subtle and do not produce immediately visible effects, just like rust that slowly forms on a car. We don't see every single atom oxidizing, but it's the cumulative effect of these atoms that produces the visible rust. Regarding the accumulation of genetic load, we have identified more than 6,000 Mendelian genetic diseases in humans. A Mendelian genetic disease is caused by a mutation in a single gene, such as hemophilia, for example, which produces a noticeable effect and can be traced through generations.
So, according to the professor, every man passes on a series of mutations to his children, generation after generation, and these are so small that they are not noticeable, but when they add up in the end, they risk compromising the way DNA functions as a whole. According to neo-Darwinists, however, to stop this drift, natural selection would intervene, eliminating negative mutations to make room for positive ones.
Personally, my specialization is in genetics, with a specific focus on plant genetics, selection, and plant improvement. Having worked in this field for many years, I feel qualified to speak authoritatively about natural selection. Natural selection undoubtedly has a role, but its scope of action is extremely limited. Although it is possible to select a particular mutation, especially a harmful one, it is difficult to identify and select the typical deleterious mutation, since its effects are often too subtle to be detected. It's like the atoms of rust on a car. They're so small that we don't notice them, and as a result, we can't get rid of them. On the other hand, it is possible to select a particularly advantageous mutation. For example, one that helps an organism adapt to specific environmental conditions. However, what is practically impossible is to select a single nucleotide in the genome, which represents only a tiny fraction of the overall genetic information. If you carefully examine the specialized literature on population genetics in recent years, you will find that many eminent geneticists are aware of inconsistencies in the neo-Darwinian theory. These criticisms, although known within narrow academic circles, have not been widely disclosed to the public or the broader scientific community, remaining almost like a trade secret. The main logical flaw of the neo-Darwinian theory lies in the relationship between mutations and natural selection. If the mutation rate is high and most of these mutations are harmful, there is no selection mechanism that can effectively compensate for the influx of negative mutations. Natural selection can only eliminate a fraction of the most harmful mutations, but it cannot counteract a high rate of negative mutations. A useful analogy to understand this concept is that of a boat taking on water. If water enters the boat faster than we can bail it out, it will eventually sink. In the same way, natural selection can only deal with a small percentage of harmful mutations, but it can't keep up with an excessively high mutation rate. Besides this, it has been found that beneficial and harmful mutations are often located close to each other on a chromosomal sequence. This physical proximity makes it difficult for natural selection to distinguish and select between positive and negative mutations.
So, according to the professor's reasoning, if we found a way to go back in time, we wouldn't find a primordial genome, but we would find a pure genome. A genome free from harmful mutations, complete and perfectly functional.
A well-known evolutionary geneticist pointed out that, genetically, we are inferior compared to cavemen. Even though he is a fervent evolutionist, he acknowledged human genetic entropy. But this is not entirely new. In fact, if we examine the cultural traditions of different societies, many suggest the idea of ancestors who were genetically superior. Ancient scriptures, for example, offer an interesting perspective on this subject. By analyzing the Old Testament, we can actually trace the longevity of the patriarchs. Starting from Noah and going through the generations up to Jesus Christ, we can observe a pattern. This pattern shows a decrease in lifespan, forming what we could define in biology as a biological decay curve. When I first read these parts of the Bible as a young man, I thought the numbers were exaggerated. But by observing the consistency of the information, you can notice a precise structure. If someone had wanted to make up such numbers, would they really have created such a precise decay curve? This pattern suggests that, over time, the writings record a real biological degeneration. Our genome is undergoing changes, with its sequences being altered due to mutations. These changes affect the functioning of our body. Unfortunately, all of us are susceptible to these changes, which affect our existence both as individuals and as a species as a whole.
With DNA sequencing, large regions have been detected that do not code for proteins and do not seem to have a specific function. These regions, called junk DNA, according to some scientists, would actually be evidence of the evolutionary history of our genome and would disprove the idea of a genome that appeared whole and fully functional.
When the genome was initially mapped, it was believed that much of it was junk DNA, a dominant concept around 2007. However, with the completion of the second phase of the Genome Project, known as the ENCODE Project, many unknown functions of DNA have come to light. The discoveries revealed that over 90% of the genome is actively transcribed, suggesting a surprisingly high level of functionality. This indicates that we need to study even more carefully to understand the functions of the remaining parts of the genome. But even more astonishing was the discovery that the genome contains multiple levels of overlapping messages. It's like reading a book where each chapter has hidden meanings in addition to the obvious meaning of the words. Incredibly, it was discovered that more than 50% of the genome is read on both strands of DNA, not just one, as previously thought. This is comparable to reading a sentence forward and finding one meaning, then reading it backward and discovering another message. This raises doubts about whether such complexity could arise through random changes. Even in the world of computer programming, every bit of code, whether it's a 1 or a 0, is there for a reason. Nothing is left to chance. In the same way, the concept of junk DNA is increasingly being questioned in light of new discoveries.
If we accept Sanford's theory and reject the idea that mutations and natural selection are the forces that have shaped and refined the genome, we are faced with an inevitable dilemma. Was the genome created by an external entity? Or did it emerge spontaneously as a result of mutations and evolution? Professor Sanford seems to have no doubts.
It's becoming increasingly difficult to explain how the genome could have formed, especially considering its multiple layers of overlapping information. Our best computer programmers struggle to create code of such complexity, which makes human technology rather rudimentary compared to what we observe in the genome.
Sanford's research and the professor's scientific stature make the theory of genetic entropy fascinating and one that raises many questions. But it's also true that it contradicts everything we thought we knew about evolutionary processes and opens up entirely new scenarios. Is man, and not only man but all life on our planet, the result of intelligent design or the product of chance? Alright, let's stop here for now, but let us know in the comments what you think, and if you like the video, subscribe to the channel. See you in the next story.